Biology.
So let's go to the next question. It's asking, state the importance of seed dormancy. So we have talked about seed dormancy, the types absolute and relative, how we can break the seed dormancy, but what now is the importance of seed dormancy? So the first point of the importance of seed dormancy, we can say seed dormancy allows for dispersal of the seed in order to avoid competition and overcrowding in the environment. So if this seed is going to be dispersed and grow over there, the other one to grow over there, the other one to grow over here. So this is going to reduce competition of light, nutrients, etc., which can be able to prevent overcrowding. Because if all seeds fall here and all of them begin to grow here, remember they are going to compete for nutrients, they are going to compete for light, they are going to compete for this and that. Therefore, seed dormancy allows time for these seeds to be dispersed to different regions so that now they may begin to grow in different areas and different regions. So apart from that, the other importance you can say that it ensures that germinations occur under favorable conditions only. So it ensures that germination has taken place only when favorable conditions have been met. Because previously, remember we said that seed dormancy can be brought, it can be brought about when there are no favorable conditions in the environment. But when there are favorable conditions in the environment, now this seed will take advantage of the environment and now begin the process of growth. So apart from that, you can say it also provides time for the embryo to mature before it now begins to grow. That is a very important point, because we see that if the embryo is mature and then someone plants that seed is going to rot, that seed is going to go bad. It is not going to grow. Because the embryo, since the embryo is not mature, the embryo cannot be able to produce the plumule or the radicle. Therefore, the seed is going to die. So here the importance is that it provides time for the embryo to mature. So apart from that, you can say that it ensures that seed avoid harsh environmental conditions such as high temperature, such as low temperature, such as wind, etc. So it ensures that seeds avoid harsh environmental conditions before they begin the process of growth. Apart from that, you can say extended viability of the seed survival being planted, because if this seed is going to be planted in the best environment possible, then it will mean that this seed is going to grow to maturity and then it's going to produce a very high amount of yield. So maybe apart from that, you can say that it ensures that the seeds germinate at the right environmental condition, and also it ensures that the seeds are able to germinate at the right weather and at the right climate. So if it's a seed maybe which grows in maybe when there is enough rainfall, so it will ensure that that seed, when the season of rainfall comes, now that is when that seed will now start to germinate. That seed will not germinate when it's time for, or when it's summer, or when in drought. So that seed will remain dormant, it will not germinate. But when it's time for rainfall, now this seed will now begin to germinate. So among others, those are the importances of seed dormancy. Because if these seeds wait for the best environmental time so that it begins to grow, this seed is going to produce high amount of yield as compared to a seed which grew in a very poor environmental condition. So question next, we are being asked also to differentiate or distinguish. So I've been told, distinguish between hypogyl germination and the epigyl germination. So differentiate between the hypogyl germination and the epigyl germination. As you can look at these diagrams, so you have the diagram of the hypogyl germination, we have the diagram of the epigyl germination, as you can see. So to differentiate between these two types of germination, we consider the cotyledons. It is the cotyledons which tell us that this is this direction to follow, this is this direction to follow in terms of definition. So we consider the cotyledons' position. Like for instance, let's talk about hypogyl germination. So for the hypogyl germination, this is the germination which occurs when the cotyledons remain buried underground as the seed is germinating. So if the cotyledons remain underground as this seed is germinating, that is hypogyl germination. But for the epigyl germination, you see that it mainly occurs when the cotyledons come above the ground as the seed is germinating. So if the cotyledons come above the ground as the seed is germinating. That is epigyl germination. Again, remember, for you to define or differentiate between hypogyl and epigyl germination, talk about the cotyledons. So for the hypogyl, remember, the cotyledons remain underground as the seed germinates. While for the epigyl germination, the cotyledons come above the ground as the seed is germinating. So we only consider the cotyledons when talking about these types of germination. So, question next we are being asked here, differentiate between hypocotyl and epicotyl in germination. So differentiate between the hypocotyl and the epicotyl in germination. So the hypocotyl and the epicotyl in germination, these also, they come from these types of germination that we have just discussed, which is the epigyl germination and the hypogyl germination. Whereby for the hypocotyl, we see that the hypocotyl lies beneath the cotyledons and above the roots. So beneath the cotyledons and above the root, that region is the one which is called the hypocotyl. So what's the function of this hypocotyl? Because you see that is where it's located, but what's the function of the hypocotyl? So for the hypocotyl, we see that it elongates during germination it elongates during germination to push the cotyledons and the shoot above the soil surface. So that is the function. Like in order to just push the shoot above the soil surface, so that they may emerge above the soil surface, so that the leaves will now be exposed to the environment to facilitate the process of growth. Also in this region, we see that this is where the roots begin to develop, at the hypocotyl region. This is also where the roots of the plant also begin to develop. But what is the epicotyl on the other hand? So for the epicotyl, we see that it is located above the cotyledons. It forms the shoot system, which includes the stem and the first leaves. That is now the epicotyl. So remember, for the hypocotyl, we see that it lies beneath the cotyledons and above the root system of the plant, while the epicotyl is located above the cotyledons, whereby this one now forms the shoot system of the plant, which also may include the stem and the first foliate leaves. Not the foliate leaves, sorry, and the first leaves. So for the next question, we can see that it's asking, explain the importance of conditions necessary for germination. Explain the importance of the conditions necessary for germination. So for the conditions necessary of germination, remember these, we studied them in primary school. We also studied them in high school in the topic of nutrition in plants and animals. So for this condition, remember we said we have water, we have temperature, we have temperature comes with warmth. Then also we have the embryo, we have the chlorophyll, we have etc. and etc. We have the pH and many more. So let's talk about the first one, which is water. So what is the importance of water in the process of germination? So as we had discussed concerning the seed coat, why it is important for water to penetrate the seed coat, remember it said that this water, as it enters through the seed coat, is going to hydrolyze the food in the cotyledon, making it available for the embryo in order to facilitate development and growth. So the first thing is that water acts as a solvent. It dissolves the food in the embryo, or it hydrolyzes the food in the embryo, making it available for it hydrolyzes the food in the cotyledon, making it available for the embryo to use the nutrients for growth. Apart from that, you can also say that water acts as a transport medium to transport nutrients within the seed from one spot to the other. Apart from that, you can say that it is through water whereby different chemical reactions take place. So the different metabolic reactions within the seed or within the plant which is germinating, that is also where in water, that is also where now most of these chemical reactions take place inside the germinating plant. Apart from that, you can also see that water softens the seed coat, thereby breaking the dormancy. Because remember, for the heat treatment, you say that we can place the seeds in warm water in order to break or make the seed coat to become soft, and then germination will take place, among others. So apart from talking about water, the next we can talk about is oxygen. So we can talk about oxygen, whereby oxygen We studied in Form 2 the topic of respiration, aerobic respiration, whereby organisms use oxygen to make energy, in order to bring energy, whereby this we say is respiration. So respiration, remember, we defined it as this is the process whereby food is chemically broken down in the body in order to release energy. So look at the previous videos whereby we discussed respiration. We went through the whole topic and we discussed the aerobic respiration, all the factors, anaerobic respiration, all the factors, etc. So in this case, oxygen is used in respiration, whereby for the plant making its own energy to facilitate the metabolic processes. So apart from that, explain the importance of temperature. So what is the importance of temperature? So optimum temperature activates enzymes. So if these enzymes have been activated within the germinating plant, so this plant is going to grow faster, or growth is going to be accelerated in this plant. So for the temperature, we can say that optimum temperature is used to activate the enzymes in order to facilitate growth for this plant species. Then apart from that, we can talk about also the embryo, whereby the embryo must be mature, because if the embryo is not mature, then this seed or this seedling or this germinating plant will never ever grow. So the embryo must be matured. So apart from that, you can also talk about the soil texture, which is the other factor which is necessary for germination. So the soil texture. So the soil must have appropriate nutrients and good drainage which facilitate the process of growth. So if we have nutrients, so this plant is going to take in the nutrients. The nutrients are going to be used for different metabolic processes within the plant, and then this plant is going to grow even faster. So if these plants grow in a waterlogged area, they point for good drainage. If they grow in a waterlogged area, so some parts of this plant are going to be in contact with water for too long, and then these parts are going to rot. So if it's the seeds are going to rot. If it's the radicle is going to rot when placed in a waterlogged region. So the drainage must be good, the drainage must be optimum in order to facilitate the process of rapid growth for this germinating plant. Apart from that, you can also talk about the pH. So the pH must be optimum. If it's basic, if it's acidic, if it's neutral. So the pH must be optimum in order for the different plant hormones or different plant enzymes to function. So if we don't have optimum pH, so this non-optimum pH is going to destroy the seed, is going to destroy the germinating plant. So the pH must be optimum in order to activate the different enzymes and facilitate the functioning of the different hormones within the germinating plant. So apart from that, you can also talk about the light. The light must also be optimum, having the most optimum or having an optimum wavelength in order to facilitate the process of photosynthesis, because light is mainly used for photosynthesis, activating enzymes for these plants or for the seeds in order to facilitate the process of growth also, among others. So those are just a few, lakini kuna zingine. So the next question, we can see here we have a diagram. You can see that's the diagram of a bean seed. That is the embryo we have been talking about. That part, that is the embryo, which you see it has been labeled as part C on the top side and part D on the lower side. That is the embryo we have been talking about. If this embryo is immature, and this seed is, it's okay, the seed is large, but this embryo is immature, the seed is never going to grow. Because the embryo is immature, it cannot be able to develop. So the seed will remain dormant. So that is the embryo we have been talking about. So this question is asking, the diagram below is a bean seed section. Name the parts A, B, C, and D. Yeah. So the diagram is a bean seed section. Name the parts labeled A, B, C, and D. So what is the name given to the part labeled A? As you can see, the part labeled A is now the outer surface of the seed. So what is the name given to the part labeled A? The part labeled A in primary school, we called it tester. In high school also, we call it tester or we call it the seed coat. So either can do. If you choose to call it tester, it's still okay. If you choose to call it a seed coat, it is still okay. So part B, that is the cotyledon, whereby, now remember we say that when water enters through this seed, it is going to hydrolyze the food in the cotyledon. Now that food is found in this region called B. So the function of water is now to hydrolyze this food, making it available for the embryo to use, and then the embryo will now begin to grow, forming the plumule, then forming the radicle, which will now emerge out of this seed. So what's the name given to the part labeled C? So part labeled C, that is the plumule, and then finally the part labeled D, that is the radicle. So those are the parts in the diagram we have been asked to label. And then we go to question B. So question B, it was asking, what is the function of A and B? So what is the function of the part labeled A, which is the tester or the seed coat? So for the tester, we see that it protects the inner tissues. This A protects part B, it protects part C, it protects part D from mechanical damage. And that is it. So it protects the inner tissues, the inner cells, the inner parts from mechanical damage. What is the function of the part labeled B? Remember we said that was the cotyledon. So the function of the part labeled B is to store food and nutrients for this seed. That is the function of the part that is labeled B. So, as you can see, this is the diagram. This is the diagram of a dicotyledonous seed that we looked in this topic of growth and development. So for this dicotyledonous seed, so you can see that we have the different parts. We have the cotyledons, yes, we have the seed coat, yes, and then we have the embryo. So for this embryo, remember we said that this is the part which now grows in order to form now the plumule, which will now form the leaves, in order to form the radicle, which will now form the roots of the plant. Apart from that, you can see we have the epicotyl, and then we have the hypocotyl, which will eventually form the roots, remember what we said, and then the epicotyl, which will eventually push the shoot up or the leaves up in order to absorb maximum sunlight for the plant to facilitate the process of photosynthesis. So this is a sample diagram of the bean that we have discussed in this topic of growth and development. So remember the cotyledon. So for this cotyledon, it contains the nutrients for the developing embryo. Like for instance, remember we said, if this seed will take up water, so the water will enter through the seed coat and into the cotyledon. Now this water function is that this water is going to hydrolyze the food which is found in this cotyledon, making it available for now the embryo to begin using this food. Now the embryo then, like it uses these nutrients obtained from the hydrolyzed cotyledons, and then it now begins to grow. The plumule forms the leaves, and then the radicle, they form the roots of the plant, and that process continues like that. So question next is asking, explain why the radicle develops faster than the plumule in germination. So why is it that the radicle develops faster than the plumule during the process of germination? So the first answer we can give here, we can say that it develops faster in order to facilitate the process of absorbing water and preparing the seedling to form a mature plant. So it develops faster in order to begin the process of absorbing water, process of absorbing nutrients, and then provide support for the seedling. So it forms a very firm support on the ground, so that now the other parts of the plant will now start growing upwards, preventing it from falling sideways. So that is why the radicle must always grow faster than the plumule. It grows fast inaenda chini, then now the plumule now emerges and now begins to go up. That is now what the question was asking us. 3 germinating seeds were placed in a petri dish containing moist cotton wool. Yeah, as you can see. So 3 germinating seeds were placed in petri dishes containing moist cotton wool. As you can see, the 3 diagrams. So we have 1, 2, and 3. So the dishes were placed in vertical position for 2 days in the dark. So they were placed in vertical positions for 2 days in the dark, as the question is saying. So state the change observed in all the 3 radicals. So state the change that was observed in all the 3 radicals. So for all the 3 radicals, we see that these radicals, they grew going downwards. That is it. So for the radicals, they must grow going downwards. For the shoots, they must grow going upwards. So the simple answer to this was that the radicals grew going downwards. And as you can look at this diagram of the seedlings which have been placed horizontally, so you can see that like on the radical side or on the root side, so the roots are going to curve downwards in order to go deep into the ground, while the shoot is going to grow upwards in order to go up towards the, we can say towards the sky. So, auxins hormones are the hormones which are responsible for facilitating such a process. On the shoot side, the auxins are going to become many on the lower side, as you can see in the diagram. So the auxins are going to be many on the lower side. Now these auxins being many on the lower side, this lower side is going to grow faster than the top side. So this lower side, remember, is going to grow faster than the top side. If it grows faster, then this lower part is going now to increase like that, because auxins are growing faster and then making the shoot part to face upwards. So that is the function of auxins that you are going to study in Form 4 in the topic of reception, response coordination, subtopic of hormones. So we are going to look at auxins. So remember, where auxins are, where auxin hormones in plants are, that part is going to grow faster. Where we don't have auxin hormone, that plant part is going to grow slowly. And that's why for the growth parts in plant, remember we say that growth mainly occurs in the meristems or in the meristematic regions of the plant. So these meristematic regions have very high concentration of auxins. So if these regions have high concentration of auxins, that's why wherever we have meristems, we have auxins, and then that part is going to grow faster. So remember we say that we have the apical meristem found on the shoot. Auxins are very many on the shoot, that's why the shoot grows going upwards. Also, the other apical meristematic region we have on the roots. Since this part, the apical meristem on the root, has very many auxins, this part grows faster going downwards. So that is the function of now these auxins, in order they facilitate the process of growth.
Biology.